Go ahead and get started. Hello, everyone, and thank you for joining the Thomas Expert Series. We're going to get started in just a minute. In the meantime, I would like everyone to please mute their microphones. This Expert Series will be recorded and placed on demand, so please let your colleagues know how much you enjoyed the presentation today. To get everyone started and make sure everyone knows where the chat function is, just let me know where you're tuning in from. Let me know what the weather's like out by you. I'm in New Jersey and it is raining cats and dogs out there. How about everywhere else? Philly is pouring. Oscar was on here talking earlier. He said it was over 105 in Florida. Colorado is nice and sunny today. Awesome. At this time, I'm going to go ahead and turn it over to Glaucos Souza, the Director of Global Business Development and Innovation, here today to talk about 3D cell culture with aspects for automation.
Thank you, Heather. Can you hear me okay?
Yeah.
Oh, great. So I'll be talking about 3D cell culture, but mostly I'm really gonna focus in what we call magnetic 3D cell culture. And as soon as I can flip my slide, hold on 1 second. There we go. So we're, as I said, we're gonna mostly talk about 3D magnetic 3D cell culture. But first, why 3D cell culture, right? We know how to culture cells. We take cells, put in a petri dish, they go to the bottom, they stick and they grow flat. But the reality is when the cells get stuck to plastic and grow in a petri dish on a tissue culture flask, they grow flat. And that's not a good representation of how cells grow in vivo. And if you're developing a diagnostic test, if you're developing a tissue model, you wanted the tissue to be predictive of in vivo biology. And today we know when cells grow in 3 dimensions, they better represent how cells grow in vivo in the body. And again, when the cells grow flat, they only have contact with plastic. They go in 3D, they have cell-cell interaction, and they have the 3D space to interact and grow more like in vivo. So I like this slide because you don't need to be an expert. And this is one of the first results we got that we published in Nature Nanotech, and kind of visually illustrates the difference between 3D cell in vivo tissue and 2D cell culture. So on the left, it's a brain cancer xenograft. We took the cells, put it in the mouse brain, grew the xenograft. And on the 2D on the right is the same cells grown in a monolayer. And the red here is encadering. Encadering is just a cell surface marker, and we're staining for it. So you can see that the distribution of the red color here is very different from the red on the 2D culture, which means the cells are behaving differently. But then we took the exactly same cells, we grew in 3D, and we did exactly same staining. And you tell me, what does that look like? That looks like in vivo. So 3D cell culture, and when we first started, we didn't have that much data that shows that, 3D is really similar to in vivo. But that's what we want. And this is what the result shows. So in 3D cell culture overlaps to just about all the areas in cell culture in general. Drug discovery, cancer, most 3D cell culture research started with cancer, tissue engineering, of course, if you're dealing with stem cells, toxicology. If you have drugs, you always want to test for toxicity. And even the FDA now has the FDA Modernization Act, which actually allows now use of 3D models to replace animal use. So it's a meaningful breakthrough for 3D cell culture. And of course, skin models, regenerative medicine, creating tissue is where we apply 3D cell culture. And there's a lot of different terms that people use in 3D cell culture. And like everything else, sometimes we make it more complicated than it needs to. So there's spheroids, organoids, there's different terminology. But generally, for the sake of what we're talking about, spheroids, we're referring to a cell aggregate. You take cells, you bring them together, cell-cell interaction take place, and they start growing as an aggregate. And an organoid is really when the cells grow from a stem cell. And they're clonogenic. They have a stem cell that differentiates and form this organotypic type of cultures. Most of what we're talking about are going to be spheroids. So now I'm going to jump into magnetic 3D cell culture. And I always like to tell where the technology came from. The technology came from a collaboration between MD Anderson and Rice University. I was at MD Anderson at the time. I developed the technology with a few colleagues, friends from Rice University. We patented the technology, we published, and then we spun off a company called Nano 3D Biosciences, where we continue to develop the magnetic 3D cell culture tools. We partnered with Greiner early on. And in 2018, Greiner acquired the magnetic 3D cell culture technology, which is why I'm with Greiner now. And as I said, sometimes we make things a little bit more complicated than they have to. And I really like this quote really resonates with me. Everything should be made as simple as possible, but not simpler, right? Einstein said that. And that's sort of the motivation of magnetic 3D cell culture. So when you go from a 2D culture to 3D culture, we all have to accept complexity. It is more complicated. But when we go from 2D to 3D, you really appreciate how important, how valuable it is cells stuck to plastic. Because when cells are stuck to plastic, it's easy to change media, it's easy to do immunofluorescence staining. And when we go to 3D cell culture, that doesn't exist. It gets complicated. So we want to make it, we need to accept some complexity, but we want to make it as simple as possible. And the way we kind of try to make 3D cell culture as simpler as in 2D is by using magnetic nanoparticles. We magnetize the cells. So we stick this, we decorate the cells with these magnetic nanoparticles. They just interact electrostatically with the cell membrane. Is non-specific, and you only need a few nanoparticles to magnetize the cells. Then you do the exactly workflow that you use, detached cells, count, and then you can place the magnetic field on the bottom, which we call the bioprinting. You have a cell repellent surface, and externally you put the magnet on the bottom, and the cells focus. Or you can put actually the magnetic field on top, and the cells actually levitate from the bottom and generate a 3D cell culture. And generally, the tool that we have, even though we call bioprinting or levitation, it's really magnets with the tissue culture plates, cell repellent tissue culture plates, and we sell as a kit. So the kit, you have your nanoparticles, you have the magnets, you have the cell repellent plates, and depending on the format, the throughput you want, you're going to have different number of magnets. So this is just a illustration of several results that we have. Because we use magnetic field, we magnetize the cells, you can bring the cells together fast. You can generate these spheroids, by promoting cell-cell interaction, you can generate 3D structures faster than most techniques. We can also co-culture different cell types. In this case here on the right, we have a glioblastoma, GBM cells, labeled with GFP. The red is normal human astrocytes. They were cultured separately, and then we use the magnetic field to bring them together. And you can see the glioblastoma, which is the brain cancer, you can see to invade or overtake the astrocytes, which, this can be used as a model of metastasis. On the bottom right here was a co-culture pre-adipocyte stem cells with endothelial cells, where when we first started this work with a collaborator in Houston, we want to generate this adiposphere, which it looked like in vivo fat, but also had the endothelial cell phenotype within the culture. So we co-culture the pre-adipocyte stem cells with the endothelial cells. The endothelial cells are GFP green, GFP label are the green cells. The red is pyrilipin, which is adipocyte marker. And you can see the sprouting of the endothelial cells within the culture. And what is important here, in going back to adding the complexity, for you to really recreate the phenotype you're looking for, which is the sprouting of the endothelial cells, we really had to titrate the cell number and figure out the right condition that you saw, that we detected the phenotype. So if you're trying to add complexity, you're going to have to do some optimization. But using magnetic cells, it really helps to the manipulation part, because it's magnets just easy to move them around and put them together. And one more thing too, this was the cover actually of the Tissue Engineering when we published this. And the reason we have this beautiful confocal image is because we use magnets also to stain. So we want to do the immunohistochemistry, put the sample on the magnet, and then you can hold them down and do all the immunofluorescence, which is not trivial to do it when you're dealing with 3D cell culture. So I like to show this slide because, again, as I said, on our journey, we started as a small company, Nano 3D Biosciences, and I like to say that Best Market is a good publication. We worked really hard to validate the technology and publish in really good journals. And of course, we also use magnetic nanoparticles. So it's a fair question. What do the nanoparticles do to the cells? And we've shown over and over in these publications that they're bioinert. We use very small amount of nanoparticle, and it's very easy to manipulate the cells with very few nanoparticles. And we have actually a white paper, a 5-page white paper that we compile the results. If it's viability, proliferation, stem cell differentiation, protein expression, oxidative stress, inflammatory stress, gene expression, with and without the nanoparticle, and the results are the same. So we have the references, we have all the results there. We also had, and I'll show some of these results, a big grant, an NCI grant with Cold Spring Harbor and Scripps, optimize your high throughput tools. And also when we did that work, we compared nanoparticle, non-nanoparticle with control cells, control compounds, and the results were the same. So if you can quickly have an idea of sort of a hands-on how the magnetic 3D cell culture works, here's a quick movie. And you have your kits here. You have your cell repellent plate. You have your magnet drives, and you have a nano shuttle solution to the kits. The first thing you do, you add your nanoparticles to your cells, and you can have the cells on a monolayer or in suspension. We have also a protocol for cells in suspension, and the nanoparticle just very easily adhere to the cell membrane non-specifically. Then you want to start your 3D cell culture. You're going to detach your cells as you always do. You're going to wash the cells with PBS, trypsinase count, and then you're going to take your cell repellent plate, put it on the spheroid drive. We call that it's a dot on the bottom of the well, and you're going to add your magnetized cells to the well. As soon as the cells see the magnet field, they will come together. I do like to show this motion because a little bit of movement at this step always moves the cells around and make sure that they all catch the magnetic field. You only need 15 minutes on the magnet. You don't need to leave it overnight or longer. You put it on the magnet, you promote cell-cell interaction using a flat surface, you generate spheroid. Now, if you want to change media, we have a bigger magnet. And the magnets are on the side of the well, so you can just put it on the bigger magnet, and the spheroids, the magnetized cultures go to the side, and you can clearly pipette the media in and out to make it easier. So it's a pretty straightforward tool. Another tool that we have on the same note of making things easier manipulation, easy of immunohistochemistry staining, in other aspects, we came up with the MagPen. And this is a tool to transfer spheroids. And I have a movie here also that will show you exactly how this works. So this is it's like a pipette, but it's magnetic to very easily move spheroids from one plate to another. So it's also, we have as a kit as well. And you have the magnets, and here we have 9624. We have your sterile cell repellent sleeve and your cell repellent plate. So you have your magnetized spheroid on the bottom of the well. Now we want to move to a different, to a clean plate. If it's to have a complete mediate exchange, so you put the cell repellent sterile plate into the well. And it's intuitive. You put your magnet in, and the cells, the magnetized spheroid goes to the top, and now you can pick up your spheroid. So you move a spheroid to a fresh plate, and your spheroids are being carried. It's on the sleeve. So you need a sleeve, because if you only have the magnet there, the cells will never come off. So the sleeve, the cells are being held on the sleeve. You take the magnet out, and we actually put the magnet on the bottom to gently pull them off of the sleeve, because some of the cells adhere a little bit, and then the spheroids just come off the sleeve. You can do a little bit of motion, so make sure they move off, and then you remove your sterile sleeve, and that's it. And you can take off the magnet, and you transfer spheroids. If you're doing immunohistochemistry, if you want a full, moving from blocking to blocking, washing your cells, going to primary antibody, you can just leave them on the MagPen and do the whole change. We also have a single tool that makes it very easy to manipulate the cultures. And here is a real result. This is not a drawing. That was a cartoon, but these are the spheroids right on the side there when you pick them up with the MagPen. Now going back to some of the things that I'm mentioning, you want a 3D cell culture to better. We're doing 3D cell culture because it better mimics in vivo biology. And this is a paper we published with Dr. Bartolomeos at MD Anderson, which they used triple-negative inflammatory breast cancer grown on PDX, and we compared the exactly cells to the PDX. So the way we did this, we took the patient cells, put it in the mouse, grew the PDX, then we took the PDX out, and we split the tumor grown in the mouse in half. In half, we broke it apart, magnetized the cells, and reassembled. In the other half, we just did the standard procedure image analysis, immunohistochemistry, and we compared the two. So we look at Icadirin, Vimentin, Ki67, and Pismad, and here is the morphology of the different samples. So this on the left is Icadirin's immunohistochemistry, paraffin embedded, both done exactly the same. But if you look at the pattern, it's very similar. The bioprinted, so the middle and the right are bioprinted structures, the left is the tumor. In the morphology, it's very close. Same thing for Vimentin and Pismad. So, and here it's a clear demonstration that, you have a tumor grown in the mouse, from patient cells, and we took the cells from that tumor, broke it apart, reassembled, and they still look exactly the same, which also shows that the nanoparticle are really not doing anything. In this paper, we also did the gene expression profile of the 3D culture. We compared the 3D culture to the mouse, and it was the same. It was 95% correlation. So, And then last set of results that I want to share is the work we did with Scripps in Florida in Cold Spring Harbor. This was work funded by NCI to validate of high throughput screening. And we actually use also pancreatic cancer organoids from pancreatic cancer cells from patients. So this is a 1,536 system. So you can see it's a very dense as far as magnets, but the left images here are just the cells in the well. And what I'm trying to show you here is that we magnetize the cells, we put in the well. If there's no magnet, the cells are just dispersed in the well. And these are colon cancer and lung cancer cell lines. But if you put on a magnet, you only need 15 minutes. The cells on a flat bottom all come together. So, and you promote cell-cell interaction and you get a 3D culture. This is also showing reproducibility, which is something very important. This is a picture of the 1,536 plate, and you can see that you have a spheroid centered in every well, and they are very much the same size. Here is another aspect that it's valuable by using, by magnetizing your cells. So when you're imaging spheroids on a flat bottom, which is usually what you want. If you want a high quality, high resolution image, you need to use a flat bottom. But when you have a flat bottom, thermal motion, usually the cells move around. And if the cells move around, often they end up stuck on the side, well, on the wall. So if you see here, the wells that have no cells is because the cells move to the 3D cultures move to the side. And that's basically, it's a simple loss because it's very hard to image when they're stuck to the wall. But if you put on the magnet for 30 seconds, you can recenter the spheroids, and not only they're recentered off the well, you know where they are, so it makes it much easier to image the 3D cultures in the well. And this is very valuable, especially when you're doing high throughput screening. Now, we're talking about 3D cell culture, and now I'm using a little bit of the different terminology that I touched on it very quickly in the beginning: spheroid, organoids, and here is a good illustration, the difference in morphology between the two. So the first two rows is H29, which is ovarian cancer cell line, pink one's pancreatic cancer cell line. And you can see we magnetize the cells on a flat bottom, bring the cells together, and after, overnight, you can see they're making these beautiful 3D structures. So the cell lines make very nice 3D structures, and they're not always perfect spheroids. As you can see, the HT29 is just about a perfect spheroid. Some cells do not make perfect spheroids, and that's just, cells do what they do. So then we took AGM1 and H21 cells. Remember, we're dealing with pancreatic cancer from patients, and these are two different patients. AGM1 is one patient, H21 is another patient. And when the Cold Spring Harbor took the tumor from the patient, they separated the cancer cells from the cancer-associated fibroblasts, which are the CAFs. So AGM1 and CAF are pairs from the same patient, and H21, CAF, and H21 are it's another patient. But what is interesting, we took the fibroblasts, magnetize the cells, bring them together, and they make beautiful spheroids. But the cancer cells, even though we magnetize the same way, bring them together, they don't form this nice, cohesive, large spheroid. They make these structures that are organoids. They are very correct. They have the cyst-like structures. They have the lumen, but it's still magnetized. You can still position them. You can still use to change media. So you have all the benefits, but they're generating organoids. Organoids are usually made in hydrogels, but in this case, we have a specialized media. Some cells form organoids by themselves, but also you can use some specialized media to generate them. Okay, great. We generated 3D structures from the patients, and part of this work, we wanted to also compare 2D versus 3D. And from the context of a high throughput screening, we want to show that or prove the hypothesis that 3D cell culture usually gives less false positives. So how did we do that? We took the FDA library. It's 3,300 compound library, and there's no cancer prescribed drugs. Therefore, if it's a drug that's killing cancer cells, but it is not for cancer, we consider that a false positive. So, and as you can see, of the 3,300 compound, the 3D with the patient samples always had less hits. So in a way, we're trying to show that we always have less false positives. And that's important because false positives can really increase the cost of a screening. You know, you don't want to have a false positive in 2D, go do all these, all the experiments, go to humans, and they fail. So we do want to screen for false positives early on in screening. So then we looked more carefully on the samples when we did the 2D versus 3D, and we plotted the results as 2D inhibitions on the x-axis, 3D inhibition on the y-axis. And as one would expect, most of the drugs fall in the white quadrant. And the white quadrant is basically these drugs did not kill the cells. Every dot is a drug. So the drugs that fell into the pink, which showed more activity towards the cancer cells, the pink quadrant are cells that were only in 2D. Then you have the overlap, 2D versus 3D, where the drugs killed in 2D and in 3D. But what was interesting, there were very few drugs that killed in 3D for the sample. And when we looked at it, the one that was most active was disulfiram. And disulfiram actually was in clinical trial for pancreatic cancer. So not only we showed the less false positives, that we decreased the number of false positives, but we also picked the drug that was only active in 3D, which means that if this disulfiram, when it was tested in 2D, it didn't kill the cells. It didn't do anything to the cells. It only killed the cells in 3D. And so that was a pretty good result. We published this in SLAS. We also published 150,000 compound screening using magnetic 3D cell culture. And if you're doing, if you care about high throughput screening, the Z prime is 0.72, which is an excellent assay. Z primes above 0.5 are considered excellent. And the last slide basically that I have with meaningful data is automation. This is a work we did with the TCAN, where we actually show here a quick movie to show that, you know, by just placing the magnet drives or the magnet array on your automation platform, you can take your plate, put your plane on the magnet, fully automated without a problem, because the magnets fit straight on the automation platform. So the robot put the plate on the magnets. Now the robot's picking up the cells. We're going to dispense the cells. And as soon as the cells see the magnet, they'll come to the middle. In this case, they left on the magnet for 1 hour. The only reason they left for 1 hour instead of 15 minutes was because the workflow was best with 1 hour, which is not a problem. So 1 hour later, they just take you don't need to leave it on the magnet. They just took the plate off the magnet and put it in the incubator. And as you can see, you generate very nice spheroids, as you can see on the bottom right. We did also screening using cell titer glow and the Z prime again well above 0.5, which is an excellent assay. So it's again, the magnetic field is just to make things easier. And people ask, why you call this bioprinting? Because you can use magnets, we can pattern. We are printing cells here at the end for nanoshuttle. So we can make it more complicated or just use for what we need. And last thing I want to show is we also in the space station, we got a cases grant, and this is Peg Whitson working with the magnetized cells in space. And this was pretty exciting. And again, we send the magnetized cells to space basically to help manipulation. It's not trivial to manipulate cells in space, although without gravity, they always go in 3D, but we still need to change media, manipulate them. And that's not trivial in space. So we just use magnetized cells to perform that. And that's my last slide, and I'll be glad to answer any questions.
Thank you so much. While everyone is typing out their questions, I do want to steal the screen real quick and just show you that Griner is running a promotion, and that is hanging out right in here, as well as a survey, literature, web links, and videos. And this talk will be on demand by tomorrow, and I don't see it right now, but there will be a little button up here that says show on demand talks, and you should be able to click on that and view this talk by tomorrow. And you can also head over into the expert series labs. Oh, I am having some connection issues, so that's probably what's going on. So you could see those as well. And I will go ahead and hand it back to Glauco to see if there's any questions.
Any questions? Should I check the chat? Hold on.
Yeah, it looks like 2 questions are in the chat.
Yes. What is the kit and how much would it cost? That's always a trick question to me, and I always defer that to Dave and to the distributor because I don't want to get in trouble. We know that that prices can vary. So and again, I'll tell you that the price is comparable to other kits. If you're using nucleic acid purification kits and other kits, it's very similar. So but contact Thomas to check the price, and we'll be more than happy to assist you. Next one is what happens to the nanoparticles? So that's a fair question. The nanoparticles, they stick to the cell membrane electrostatically. It's polylysine, very similar to how cells surfaces are modified, and we showed this on Nature Nanotech and a few other papers. So the nanoparticles stick to the membrane, and after 5 days, they're stuck in the extracellular matrix, which means they don't go anywhere, which they don't do anything to the cells, and also the 3D cultures stay magnetized, so it makes it much easier to manipulate them without adding more nanoparticles. So let me see. There's one more. Does the system work with all cell types? As far we have tested dozens of cell types, and they pretty much have worked with just about any cell type. However, I do tell people, if your cell is very difficult to grow and is very delicate, we don't do magic, right? So we can help. We can push the cells together with cell-cell interaction. We've seen that it does help co-culture different cell types. But as far as we've done, from stem cells, neurons, cancer cells, primary cells, liver cells, kidney cells, again, primary stem iPSC cells. So we have a very long list, and most of these are published either as a peer-reviewed publication or in an application note.
Anyone have any other questions? You can come off mute if you have any other questions as well. All right, I don't think there are any more. No one is speaking up. So thank you so much, Glauco, for joining us.
Perfect.
Enjoy the rest of your day, everyone.
Thank you very much.